Assalamu alaikum class and respected teachers. Myself, Isha Ijaz, and my partners are Mariam Abid and Hafiza Maham. Today, I will be going to present the topic of DNA barcoding. In today's video, I am talking about what is DNA barcoding, why it is important, how DNA barcoding works, and its major applications. We need biodiversity to survive. But now, over 1 million species are threatened with extinction, and threats to biodiversity results in crises like new diseases, invasive species, and inconsistent food production. Major research programs use molecular methods such as DNA barcoding to track biodiversity on a plant-century scale. Together we can build a baseline of biodiversity data so we can measure changes due to climate change and anthropogenic activities and protect our biodiversity. DNA barcoding is a method that uses an organism's DNA to identify it as belonging to a certain species. In Canada, our scientists are at the leading edge of DNA barcoding at Agriculture and Agri-Food Canada. We have been identifying thousands of good and bad organisms and registering their DNA barcodes to international data bank. The benefits of this working with DNA barcodes are huge for our agricultural crops, livestock, and trade. Now let's see how DNA barcoding is done. DNA barcoding involves 5 major steps. First, a little biological sample of an organism is collected from which the DNA is extracted. After this, utilizing a specific primer set, few hundred bases long-term target region of nuclear mitochondrial chloroplasm genome is amplified using PCR. The target region chosen for a PCR amplification depends upon the organism's type. For example, the 16S ribosomal RNA gene is the target in bacteria. For fungi, it is the internet transcript space region or ITS region. RBCL gene of chloroplast and METK gene of mitochondrial DNA are suitable for developing DNA barcodes in plants. In animals, the segment of mitochondrial gene cytochrome C oxidase serves as the target region for 10-upping the DNA barcode. A set of universal primers are used to amplify one or more of these target regions for PCR amplification. These primers are conserved to work in all of these species of a particular taxonomic group. The amplified DNA is then purified, cloned in a vector, and sequenced using sequencer to obtain the exact sequence of target barcode region. The resulting DNA sequence is compared with all the non-DNA barcode sequences of all these species in an existing reference database. If the sequence is a perfect match, we identify the unknown species. If some DNA variation is found in the DNA barcode of our unknown organism, then it may be a novel species. Now let's see some of the useful applications of DNA barcoding. With DNA barcoding, one can quickly and accurately identify the species of unknown organism, even if it is difficult or impossible to do so, based on its morphological appearance. DNA barcoding is helpful in estimating the biodiversity, species richness, speciation, and evolution by comparing the DNA barcodes across different species. DNA barcoding also has its application in conservation over time. Distribution and existence of a particular species may change due to the human interference, climate change, or any other. DNA barcoding can be used to monitor changes in the distribution and abundance of different species over time. This information can be used to identify the areas that are particularly important for conservation. DNA barcoding can also be used to verify the identity of food products, check if the products are properly labeled and free for contaminants and adulterants. So today, we have learned what DNA barcoding and how using a small DNA sequence we can identify, classify, and characterize species and organisms for various applications. Thanks for watching.